Morning. Welcome to Biology 1A. The lecture is simulcast over in 10 Evans. So if you want, there's plenty of chairs over in 10 Evans. Lecture's webcast there. The syllabus is there as well, okay? So please read the syllabus. It has very important information for you. Discussion sections do not begin this week, nor do labs begin this week. Both labs and discussions begin next week. Okay, again, I'm going to say that one more time. Labs and discussions begin next week. You don't need to go check into the lab locker this week or anything like that. Everything begins next week. You must go to your assigned lab and your assigned discussion next week. If you don't show up to your assigned section, we will end up dropping you, basically. As you can see, the class is very full. There's also people in 10 Evans. So we have over 600 students enrolled, about 650. This room holds 500, so we will need to use 10 Evans. With respect to materials, so we're using the Russell Biology book, the second edition. We are not going to use Aplia, so it turns out the faculty decided not to use it for points. Instead, what will happen is each faculty member, Dr. Malkin will teach the first third, Dr. Ernoff will teach the second third, and Dr. Firestone the last third. Each faculty member will get 1 point per lecture, so effectively about 13 points. Dr. Malkin and Dr. Firestone will have those points on the final, so there'll be extra points on the final. Dr. Ernoff will use iClicker questions, so we expect, at least if you want the points, to attend lecture 8 to 9 when he's lecturing. We'll have it in 10 Evans as well, and we'll use the iClickers. You'll need to register them, and that's how points will be awarded for his part of the course. Any questions about that? With respect to the book, it does come bundled with Aplia. We are not going to require it. It's a useful resource. The book is highly discounted. Okay, so right now at the bookstore, it's $104 for a new book. It comes with ebook plus Aplia. Aplia has, various assignments, quizzes, things like that. So you can use it as a study resource. It has animations, etc. So I recommend it, but it's not required for points. Now that $104 at the bookstore is cheaper than the cheapest used book on Amazon. So I managed to get about a $50 discount for the bookstore. So I do recommend buying it new, so you get the ebook and the Aplia. However, if you can find it from a friend, buy a used book, things like that, just be aware that the price at the bookstores are highly discounted at $104. Any questions about the textbook? Okay, Russell second edition. It's identical to the second edition. The UC has a custom cover. That's how I managed to get the $50 discount. With respect to, yes, question. I'm sorry? So Aplia is available for purchase separately. I think it's $90 separately and $104 for the book, ebook, and Aplia. Okay, I got a major discount on it. So any other questions about that? Other materials you need for Bio 1A. I mentioned earlier in the B-Space announcement, you'll want to have Dr. Malkin's reader during lecture. He will refer to figures as we go through. Dr. Ernoff will post his lectures on B-Space, and Dr. Firestone will have a reader as well. There's also an exam reader which has past exams from the faculty. I highly recommend you purchase it and use it the correct way. Now, I've been teaching this course for about 20 years. Dr. Malkin's been doing it 20 years as well, even more. So we enjoy teaching this course. Students are great. You study really hard. But the biggest issue is students fall behind. So last semester, we had Aplia assignments to try to keep students current with material by having weekly assignments. We're not doing that this time. So a little bit more burden upon you to be responsible. Do not fall behind. That's the biggest issue. You will not be able to cram for this class the night before the exam. So the way you want to use the exam reader, several days beforehand, set a timer for 50 minutes. Set a timer for 50 minutes. Set aside the time. Take the exam. When the time is up, you must stop the exam. If you didn't get through it, that's a way to indicate your pace. If you're going too slow, you're going to need to pick up the pace for the actual exam. Then go back and grade it. If you got a 98, you can probably feel pretty confident that you're at the right level for the actual exam you'll take. If you get a 60 or 50 or 70, if you're not happy with it, then focus on the areas you missed. Did you miss mostly metabolism? Did you miss mostly the cell stuff? Use it as a study resource. Okay, you can always abuse it. Just go through, oh yeah, I thought that was the answer. I didn't really bother to check. Oh yeah, it's either C or D, and yeah, I thought it was C, and yeah, it's C, okay? So again, the resources are there. Please take advantage of them. Exam dates. You'll find out more on Monday when the lab lecture is held. The lab exam dates are the 14th of March. That's a Wednesday night. That will be lab exam 1. The second lab exam is held the last week of lab classes, the 23rd through the 26th of April. In this class 1A, exams will be 8 to 9 for the first exam on February 17th, and the second exam, March 23rd. That is the Friday before spring break. So mark that on your calendar. Friday before spring break, you have an 8 to 9 exam. If you're taking off early, make sure you don't take off until after the exam. Any questions about that? All right, and then of course the final will be posted. Enjoy the class. My name is Mike Meehan. My information is on the syllabus. Dr. Malkin will have a few comments about lecturing and things like that as well.
Sure. Thank you. Okay. I'll see you at 10, right?
Okay.
Can you hear me? No. Bad. How can I hold the microphone and talk at the same time? Okay. Let's see. God, it has to be way up. Yeah. Hi. Good morning. My name is Dick Malkin, and I'm the first lecturer in this class. And I've given you some pertinent information about how to get a hold of me. I am a professor, and please note emeritus. What does that mean? It means I'm retired, okay? And I've taught this class for 20 years, over 20 years, and like 7 years after I've retired. And everybody always says, why are you crazy? Why are you teaching a class that has 600 or 700 students when you're retired? And my answer is twofold. One is I really like the material I talk about in this class. I think it's very exciting, one of the most exciting parts of biology today. And I really like the students in the class. A lot of fun, challenging, often crazy, but those are the things that I've enjoyed. I've always taught it in the fall. This is the first time I've taught spring semester. And it's cold out at 8 o'clock in the morning. In the fall, you know, you just wear a shirt, and it was sunny and bright, and God, it's dark and bleak and cold. So everybody's all buttoned up, okay? Okay, so that's that bit. And I'm in the plant and microbial biology department, which is in College of Natural Resources. The other lecturers in this portion of the class are from MCB. My office is in Koshland Hall. My email is there. Much of this is in the syllabus, and my phone number is there. I will have office hours after lecture, Monday, Tuesday, Wednesday. I'll take Thursday off, might add it on later, and Friday, okay? The office hours are held in 2084 VLSB. That's right across from the teaching labs that you'll be going to. And you should become familiar with that room because lots of things go on in there, and there are office hours. My office hours are there. There are GSI office hours there every day from 10 until 2 p.m. I want to point out that we try to make ourselves accessible to you. I try to, at least I can't speak for the other faculty, but I like to try to make myself accessible. That is, I've got 4 or 5 office hours a week. I'm pretty good about answering email, especially now since I'm not burdened with administrative and research stuff at the university. And for those who are interested and are energetic, I go to the gym every morning at 6 a.m. when it opens. I was there from 6 to 7, and then I came here. And on occasion, students will come up to me and say, "Oh, Dr. Malkin," you know, and that's fine. That's fine. That's great. Okay. The other thing is, when I leave here at 9 a.m., I walk down to LSB. You're more than welcome to accompany me down to LSB. The lectures, I want to point out, start at 8:10, and I always hold to that. And they have to end at 9, because I don't know if you know this room. Sure, you've had chemistry in here probably. There's this rotating table, And at 9 o'clock, the chemists run in here and they start rotating you away. It happens, believe me. So, and the chemists are very aggressive. So we have to get out of here by 9 o'clock. Okay, a couple of comments about the course. Mike has mentioned the syllabus. Please read it. There's some good information in there. He did not mention that we webcast lectures, and those lectures are not simulcast. They come up on the web, I don't know when, a few hours after the lecture. And the effect of webcasting the lectures, we have been webcasting for a number of years, is that you look around the room today, there's over 500 students here today. On Friday, there will be 400 maybe. And as the semester goes on, fewer and fewer students come to lecture, and more students watch the webcasts. And we who teach this, I think, believe it's important to come to lecture. And we believe students that come to lecture do better than students that simply watch it on the web. The other thing the web does is something Mike referred to, is it tends to make you put off studying the material until the weekend or the few days before the midterm, and that is the kiss of death in this class. There's a lot of material. We go through a lot of material, all of us. We go through material very quickly, and I think if you believe you can cover my 13 lectures or 14 lectures in a few days before the exam, you are sadly mistaken. So what I recommend is that you come to lecture and you look at the webcasts of lectures that are causing you problems, okay? If you're confused or don't understand something, make it a secondary resource for you. Don't make it your primary resource. No one will listen to me. This yellow book, canary yellow this year, very nice, is essential for my lectures, okay? This contains about 100 to 120 figures, and I lecture from these figures. These figures are also on BSpace if you don't want to invest the, whatever it is, $7, something like that for this. I think it's nice if you have this, you can write on these pages and whatever, but I think the figures that I am referring to are very complicated, often complicated, and they're difficult to draw on the board. I'm not a great believer in PowerPoint. I like using chalk and the board. But I'll talk about individual figures in this handout. And when I sit down and make my exams up, my midterm and the questions for my final, this is where I start. So when people ask me, what do I have to know? Well, you have to know at some level what's in this handout, okay? So that's very important for you to realize that. Let's see, what else do I want to say? I won't talk about grading because that's described in the syllabus, and I think that's all I have to say about the mechanics of this class. Are there any questions? If at some point in the lecture you guys in the back can't hear me, you're going to have to wave or scream or something, okay? Because it's hard to know if you can hear or not. It's also very hard for me to see in here because the lights are very bright. But so if you do have a question during lecture, jump up and make yourself very visible maybe. Okay, so my portion of the class deals really with sort of a portion of biology that, as I said, is very exciting. It deals with biological molecules, their structures, their functions, and cell structure and cell function, and some basic aspects of metabolism. Now, biological molecules, that area, structure and function, over the last, let's see, I got my PhD, I won't tell you when, PhD from Berkeley, a long time ago. And in those days, we knew very little about the structure of biological molecules, like proteins. And that's roughly 40 years ago or so. There's been an enormous growth in our knowledge of structure and function of biological molecules, and combining that with the techniques of molecular biology, modern molecular biology, we now have the ability to manipulate systems in a way that was unthinkable, say 40 years ago. So this is a very, very quickly moving area. We have a lot of detailed knowledge. And basically what biology has done over the last 100 years, it's moved from sort of an observational science where, you know, you'd go out into the woods and you'd look and say, ah, this is a pencil. And you draw a picture of it and put it in a book. We don't do that anymore. We can get down to the nitty-gritty of what's that pencil made of. You know, what are the different parts of it? What are the molecules in there? The atoms. You've got certain thing at one end, certain thing at the other end, the whole function of this thing. And we can describe that in many cases in molecular detail. And that has given biology and biologists an enormous set of tools to approach problems in a totally different way. Okay. So I'm going to start off talking about biological molecules, their structure and function. But before talking about biological molecules, I want you to look at the first figure, which is figure 2, under how to do well, if you have the thing. It's a very simple figure. It shows the kinds of molecules that are found in cells. And the important part of figure 2 is that regardless of the kind of cell you look at, a plant cell, an animal cell, a bacterial cell, 70% of that cell is water. So the most important biological molecule is really water. And water is unique. I say a couple of words about water, and then we forget it for the rest of the semester. But you should realize that we are basically water. And all of the biological processes, metabolism, et cetera, that occur in cells is occurring in an aqueous environment. If you look at the structure of water, it's not quite as simple as one would imagine. Since I'm going to have to change my decor because I have nowhere to clip this microphone. So, well, okay, I'll try it. I don't know. I'm going to the Madonna mic. Boy, that sounds great.
No, not right. Screen.
Okay, can you? Yeah, I can hear me. That's better, right? Good. This gives me hands. Great. I've got to be careful of I can't see, though. Can't see. What? Evolution, yeah. Okay, so I was talking about water. Basically, although water has this very, very simple structure, H2O, everybody knows what water is. There's more to it. There's an oxygen atom, which has a partial negative charge, and there's two hydrogen atoms, which have partial positive charges. So that really looks bad. Sorry. Plus, minus. First lecture. Okay, so water's a polar molecule. And this allows water to form hydrogen bonds with other water molecules. And this oxygen, I won't draw what's shown in figure 3A and 3B. This oxygen can interact with hydrogens on another water molecule. This hydrogen can interact with oxygens on different water molecules. So you get this lattice structure of based on hydrogen bonds, OH bonds. Hydrogen bonds are very weak bonds. For example, if we look at bond strength of things you might be more familiar with, like OH bond, the bond strength of that is 110 kilocalories per mole. These are covalent bonds, CH. These are all roughly 100 kilocalories. In contrast, weaker bonds, such as hydrogen bonds, have bond strengths of roughly 5 kilocalories. Charged interactions, plus minus interactions, are of the same level, 5 kilocalories. And things called hydrophobic interactions are also in the range of 5 kilocalories. So what this means is these bonds can be readily broken and formed. And this is very important in the structure of biological molecules. These bonds, obviously, require a great deal of energy to make and break, and they're much more stable than the so-called weaker bonds that are found in, both kinds are found in biological molecules. The polarity of the water molecule is important in its properties as a solvent. Water is a great solvent, and that's why, everybody believes life, certainly life on our planet, is related to the presence of water. And the idea is that if we go to some other place out there in, the ether or something, there may be water, which may lead to the discovery of a life similar to ours. I think that's sort of speculative. But you have two kinds of interactions that you can have with water. You can have molecules that dissolve in water, and they would be molecules that are generally polar. They have oxygens, they have nitrogens, things like that. Ions, sodium chloride, will dissolve because, again, it can interact with the polarity which is found in the water molecule. And these molecules are known as hydrophilic. That's why I don't like this, because it falls off. Going to have to get used to it. Walk around like that. There are molecules, however, in biological systems that do not. I have to keep my glasses off, so there's a trade-off here. There are molecules in biological systems that are not water-soluble. They don't dissolve in water. We'll talk a lot about some of these, particularly molecules that are found in biological membranes. These are molecules which are known as hydrophobic molecules. And I will be talking off and on about hydrophilic molecules, hydrophobic molecules. So these are two terms that I want to introduce when I just briefly talk about the structure of water. Water has a bunch of other properties that are fairly important in physiological processes, its cohesiveness, things like that. I don't want to talk about that. It's not relevant to my topics that I'm going to talk about. But hydrophobicity and hydrophilicity are relevant, so I just like to introduce them in terms of the structure of the water molecules and how other molecules interact with water molecules. Going back to this figure of the cell and what's in the cell, if you take out the water of the cell, which is 70% of its weight, essentially, volume weight, you're left with biological molecules, the true biological molecules. And the figure shows that approximately 70% of a cell is made up of protein. Approximately, maybe 10-12% of a cell is made up of lipids. Approximately 5 to 8% of a cell is made up of nucleic acids. And then there are other molecules which are present in lower levels, carbohydrates, ions, metabolites. And interestingly, that ratio, those numbers also don't change significantly if you look at a wide variety of biological systems like bacteria, plants, animals, fungi, anything. You always find proteins being very prominent and other molecules being less prominent, let's say. Can I Okay, the four major groups of molecules that we'll be talking about are shown in figure 4. They are sugar. Instead of fatty acids, you could say lipids. Amino acid nucleotides. These are building blocks. These are building blocks which are used to produce high molecular weight compounds. Macromolecules is the general term that's used. So sugars, simple sugars, are polymerized to produce polymers of sugars, things everybody knows about. Cellulose, for example, very common carbohydrate polymer. Fatty acids and various forms of lipids, they don't produce exactly the same kinds of polymers that we're going to talk about with the other three compounds, but they do produce structural organizations such as membranes, which are based on their chemical properties. Amino acids, again, low molecular weight compounds. We'll talk more about amino acids in a minute. Amino acids generally have a molecular weight of about 100, and you'll take 1,000 or 5,000 amino acids and you put them together. You'll build a polymer by stringing amino acids together to get proteins, and the same sort of system holds with nucleotides, low molecular weight building block compounds which are put together to synthesize nucleic acids. So biological molecules, all of these polymers follow a very similar sort of rule where you have a low molecular weight compound and then you polymerize that to form a high molecular weight compound. And the high molecular weight compounds are the primary workers in the cell. You know, one thing you find very quickly in biology, I think it's an interesting point, is that nature biology in particular is very conservative. By that I mean when something is found that works, it's used over and over and over. That makes it easy for students to learn some of these things because you don't have to learn 65 different ways of making a polymer, okay? You make a polymer in biological systems a very simple way, shown in figure 6. You take a sugar and a sugar, and these have OHs, and these have OHs, and you form a disaccharide, a disugar, which has water, H2O split out. So this has a bond that looks like that, and water is released. That's a condensation reaction where water is the product, and two sugars are now locked together. This is the same basic mechanism that occurs in protein synthesis and in nucleic acid synthesis. There's always a condensation of two monomers, loss of water, and you get a dimer. And you continue to do this up to high molecular weight structures. This is a reversible reaction in the sense that when these polymers are degraded, and they are degraded in cells, they don't last forever. Proteins don't last forever. High molecular weight carbohydrates like amylose and amylopectin and glycogen don't last forever, and they're broken down at various points of the times in the cell's life, and then water is used during this process to produce monomeric units. There are many, I skipped figure 5 because it's very abbreviated and very, doesn't give you anything like the detail of information that's available on the macromolecule, particularly the examples of macromolecules, for example, proteins, which we're going to talk about. The functions of proteins are so numerous that I used to have a whole page of protein functions, but I think the book describes protein function, and I think you can, if you think about your body and your cells, most of the things you recognize rather quickly, antibodies, breathing, hemoglobin, these are proteins. So proteins are involved in pretty much all of the basic processes of a cell, And it doesn't make any sense for them to list 3 different things because there are probably 300. But this table summarizes the macromolecule, the basic subunit, which is important, and some functions, and gives you an example. We are going to go through these 4 groups of macromolecules in some detail. We're going to go through, start with proteins and then move on to lipids, carbohydrates, and very, very briefly, nucleic acids, because nucleic acids are not a major focus of my lectures. They are covered extensively in the second part of the class, which is molecular biology and genetics, where the focus there is on information transfer. Okay, what about proteins? How would one justify talking about proteins first? Well, I mentioned already that proteins represent the major biological macromolecule in the cell. Roughly 70% of, if 70% of a cell is water, but if you take the water away, 70% of what's left is protein. So they're the most prominent macromolecule in a cell. They are also, I think, the most interesting. I should mention, in my long career as a Berkeley, or short career, depends how you look at it, long or short career as a functioning professor at UC Berkeley, I worked in the field of photosynthesis in plant biology, and my interest within photosynthesis was based on work I did as a graduate student, where I studied proteins that contain metal ions, particularly copper-containing proteins and iron-containing proteins. So I am, by training, a biochemist, and I know a lot about photosynthesis. Don't know much else about other parts of plants, which has always been an embarrassment. Well, I was chairman of the department for 5 years, and people would call me up and ask these questions, and I never knew the answers. You know, why are my plants growing? This is what people do. Or if I play music to my plants, will they grow better? I mean, you know, you get a lot of strange questions as a chairman of a department. And I'd always say, well, I have to refer with somebody who's a little more, has more expertise in this field. Some of my colleagues did. Okay, so we're going to talk about protein structure and protein function. And we're going to go into a fair amount of detail because, as I said, if there's one thing we sort of understand very well in modern biology, I think it's protein structure and protein function. And we're going to really not get down to the details of every atom in a molecule, but we can have that capacity. The building block of proteins are amino acids. And amino acids are shown schematically in figure 7. You've got a carbon with a carboxyl group on it, a hydrogen, an NH2 group, and something called R, okay? And I always put in figure 7 just because people encounter a term that in books and things that I think requires definition. The term is Zwitterion. I think this is German. And basically what that means is this amino acid, and we'll talk about R in a minute, can exist in various ionized forms. You can lose this hydrogen, so you have this, or you can gain a proton, so you have this, and this is the Zwitterion form because you've got a positive charge on one end of this molecule and a negative charge on the other end. Obviously, this is going to be dependent on pH. I haven't talked about pH, but everybody here has had chemistry and they know what pH is. If you don't, you should go back to the first page of this, the chemistry checklist, and see if anything on that page makes sense to you, because pH is very important in these lectures. So you might have this form of an amino acid, NH2, with the charge there on the carboxyl, etc. So as you change the pH, you change the ionization of the carboxyl and the amino end of the amino acid, And figure 7 shows the various forms that can exist. And these, as I said, these changes in charge on this simple compound, even when it's in a protein, are very important. Okay, all proteins are built up of amino acids. There are probably thousands or tens of thousands of amino acids that exist in the world, not necessarily in biological systems, but only 20 amino acids are found in proteins. So we're lucky, okay? You might say, first question I get, always get, and it's related to figure 8, which are the structures of the 20 naturally occurring amino acids, is do we have to memorize these structures? And the answer I have given for a large number of years is no. You don't have to memorize any structures of biological molecules in this class. This is what you do in biochemistry 102, okay? Or 100. When I took it was 100. Everybody took 100. And there was no 102. You really had to learn these structures. So what are these 20 amino acids, and what's that? How do you relate that to this general structure? Well, this R is a side chain in each amino acid, and there are different R groups that confer different properties on these amino acids. And the figure 8 has grouped amino acids by similarities of side chains. For example, the first group at the sort of top of that are amino acids that are positively charged arginine, histidine, lysine. So our groups here are positively charged because there's a nitrogen group in that side chain. And then there are also negatively charged amino acids, such as aspartic acid and glutamic acid, where you've got a carboxyl group in that side chain. So these are the things that are important for you to realize, is that these R groups are going to confer on that amino acid differences. And this difference in charge or lack of charge in an amino acid side chain is going to reflect itself ultimately in the structure of a protein. So the three groups are hydrophilic side chains with charges, hydrophobic side chains. Now remember what I said by hydrophobic, about hydrophobic. Hydrophobic molecules are not readily dissolved in aqueous solutions. If you look at the hydrophobic amino acids, you see very few, if any, charges. You see a lot of carbons. You see a lot of methyl groups. So this is all relative. Amino acids, in general, do dissolve in water, but the hydrophobic side chain amino acids are less soluble in water than the charged amino acid side chains. Okay? So that's what's important. When we take two amino acids and we stick them together, shown in figure 9, where you have a glycine and alanine, and you do this condensation reaction where an NH2 on one amino acid side chain reacts with a carboxyl group on another amino acid side chain, and water is split out, what you form is a bond that looks like this. So there's the carboxyl, and there's the amino group, and this is the rest of the amino acid, and this is a peptide bond. And then, of course, this amino acid has a free amino group, and this amino acid has a free carboxyl group, and you continue to build these up. And they don't build from both ends, but I don't talk about that, okay? Because you learn how proteins are made, actually synthesized, in the second series of lectures. So for our purposes, we have proteins which are made, contain peptide bonds, and we will not deal with how these peptide bonds are actually synthesized in the cell. Now, you continue to go up, and you make proteins. Which have hundreds and hundreds of amino acids. Now, people always ask this question, and there's no answer to it. How many amino acids do you need before you can call a protein a protein? I don't know. I mean, 50, 100? 100 certainly. Well, what's the lower limit? I don't know what it is. Maybe it's 10. What's the difference between a polypeptide and a protein? Hard question to answer. We're talking about semantics, not structure. Every protein is a polypeptide, but there are some polypeptides which are, may contain 5 or 7 amino acids and are not generally called proteins. But it's not an issue to worry about. I want to make an aside here. This is an aside. An aside is when I talk about something that's, I think, interesting, but it's not important in terms of your life as a student or in this class. There is one dipeptide. Proteins are obviously very important, but there are compounds, well, I don't have to draw the structure. Aspartyl phenylalanine. Okay, aspartyl phenylalanine is a dipeptide where you've got an aspartic acid linked to a phenylalanine. How many people have heard of aspartame? Fair number, right. You know what aspartame is? That. Essentially that, slightly modified. Aspartame is a derivative of aspartyl phenylalanine. Aspartame is 200 times sweeter than sucrose. And why is this important? Because artificial sweeteners are $1 billion business in this company, okay, in this country. There are lots and lots of compounds now that are used as substitutes for sugar, natural sugar. And, saccharin was the first one. Aspartame is now very common. And somebody is obviously making a lot of money. And people are also looking for compounds that are even more sweet than sugar. So you would use less and less of them. So I don't have to tell you that you can make money in biology. That's maybe not a surprise anymore. When you make proteins by stringing together amino acids, remember, every protein has at this end an NH2, and at this end a COOH. So there's a carboxyl end, and there's an amino end. Okay? Now, we're going to look at structure of proteins. We've got a few minutes, and I can go through this and probably come back to it on Friday. When we look at the structure of proteins, there's a hierarchy in structure. What do we mean by that? Well, I mean, we start off talking about something which is known as primary structure, secondary structure, tertiary structure, and quaternary structure. And this is important. You have to understand what each of these are and how they fit into the general picture of protein structure. So pay attention to the next 5 minutes of lecture if you've been snoozing up till now. Okay, the primary structure of a protein, very simple, is the linear sequence of amino acids. That is, every protein has a unique sequence of amino acids. Some proteins have all 20 amino acids. Some proteins don't have some amino acids. Obviously, a protein that has a molecular weight of 10 or 20,000 has, 15 glycines and 10 of these. And so there's not a single amino acid that makes up a whole protein. The linear sequence of amino acids, where does that come from? This is just fact. I'm not going to describe the mechanism in detail. The information that is in a DNA molecule is converted to information in an RNA molecule, and that is converted into a protein. This is the central dogma of modern biology, DNA to RNA to protein. So there's information in the gene sequence within DNA that ends up in the structure of a protein. And that's the linear sequence of amino acids. The secondary structure, localized organized regions. What do I mean by that? It means within protein molecules, there are areas where the structure, the amino acid sequence, allows there to be organized structures. And the two kinds of structures that can exist here are known as alpha helices and beta sheets. I cannot believe I don't have a figure of an alpha helix. Well, it's in figure 14, okay, jumping from 11 to 14. You can see that there you have a primary structure of amino acids, and then the second structure in that is a helix, which is formed because of the arrangement of specific amino acids in a region. So this is why I use the term, the important term here is localized. And how many people have heard of Linus Pauling? And you all have heard of him because he pushed vitamin C, right? That's why most people know Linus Pauling. But Linus Pauling was probably one of the greatest chemists of the last century, and he predicted and came up with the idea about this alpha helical structure in proteins, and that's why Pauling got a Nobel Prize in chemistry, because his ideas on protein structure were far ahead of everybody else's, and he proved them. Okay, so you will have a protein. Here's a protein. And within that protein, you may have a helical region here, which looks like that, and you may have here a region where there are hydrogen bonds going like that. That is beta sheet. The stabilizing feature of alpha helices and beta sheets are hydrogen bonds. And some of the bonding is shown in figure 11, where you can see there are hydrogen bonds from a chain, partial chain in the protein going in one direction, possibly, and then it's going back in the other direction. So the hydrogen bond that I talked about initially becomes very important in this so-called secondary structure of proteins. Now, not all proteins contain alpha helices and beta sheets. This is another thing about these lectures. Everything I say, there's an exception to. So what I think we have to do in this class is we've got to paint with a very broad brush and present stuff to you like it's always true. But it's certainly not always true that all proteins have alpha helical regions or beta sheet regions. But they're very prominent in most protein structures. Tertiary structures are interactions. They form because of interactions between side chains. I erased those side chains. Too bad for me. Bad thing to do. If you look at figure 10, you can see there are three different kinds of interactions that are shown in figure 10. The first one, I won't talk about the first one. I'll come back to that on Friday. Hydrogen bonding. Hydrogen bonding between carboxyl groups, which are in side chains, and amino groups, which are in side chains. Those hydrogen bonds are different than these hydrogen bonds. These hydrogen bonds are related to peptide linkages. So there's a distinction between hydrogen bonding, which is involved in tertiary structure, and hydrogen bonding, which is involved in secondary structure. If you look at figure 11, you can see all the hydrogen bonding there comes from the CONH of the peptide bond. Remember, this is the peptide bond. So you can have a hydrogen bond there, and you can have a hydrogen bond there, and that is what forms secondary structure. When you get to tertiary structure, you're now dealing with R group interactions. plus some other important interactions that I will take up on Friday. Okay, I'm going to stop here because I don't want to get the chemists upset. We'll go through more structural aspects of proteins on Friday. We'll talk about quaternary structure and then some other interesting features and talk about the actual structure of some of the molecules and how they come about. How do we get them?